So the very first system that we cover in this class is skin. So there is going to be a lot of vocab information, and then you'll be expected to take those vocab words and use them to help you critically think through some questions. And so the exams do get a little more difficult as the semester goes on. And so this one is very bulky. So if you've not yet mastered how to study, you may want to use this time to really begin to go over them. You should be looking at the chapter. If you don't get all you need from this presentation, you could use the end of the chapter questions. There are homework questions that you could use to help you study. And then, of course, you can always ask me questions or go down to the Learning Center to ask them to help you. And so let's just start with skin. Skin is what we call an integument. And so we call this the integumentary system. It's the largest organ, and it has certain features branched off of it that we call appendages. So we're going to learn not just the skin itself, but the organs that make up the different, or I'm sorry, the cells that make up the different tissues of the skin organ, how the skin grows, how it repairs, and then learn a few things about hormone production and a few terms that have to do with damage and burns. So the skin is your largest organ. It measures, if we were to, it's kind of morbid to think about, but if you were to take the skin off and stretch it out, it would measure around 2 meters, which is around 20 square feet. So it's pretty large. It is our complete outer boundary covering our entire body. The integumentary system itself is made up of not just the skin, but all of its appendages. When we get into the skin, we can classify it into 2 different layers: the epidermis, which epi means above, and dermis. So it has 2 primary layers, epidermis and dermis, each made up of their own layers of cells. And then below that is the hypodermis. The hypodermis, hypo means below, lies directly beneath the dermis and allows that attachment between the fascia and the muscles. And so it's what allows your skin to not just be loosely moving around, but actually attached to those under muscles. Not all skin is the same. You have certain areas of the body that are thick and certain areas that are thin. Generally speaking, thin skin has hair and a smoother surface, whereas thick skin, like the palms of your feet, the palms of your hand, or the soles of your feet, usually have no hair. And if we could zoom in, have more of a ridged surface. Because they do different things. If you have a different structure, you have a different function. So if we could zoom in on the skin, we're going to, does anybody notice what this is? Can you see the skin here? If we zoom in, you see the stratified layer and this fascial layer here, the hypodermis, connecting it to the smooth or the skeletal muscle below. We're going to learn the different layers of the epidermis and how those cells sort of migrate upward as they mature. And so in thick skin usually does not have hair, so you're not going to find hair glands. You're going to find a thicker outer layer of these keratinocytes or keratin-filled cells that form these dermal ridges. These dermal ridges allow us to, grip, allow us to have more protection in those areas of the body that are going to, if you think about it as far as, in nature, how that works, areas of the body that allow more contact, areas of the body that need more protection. And so the thick skin has these, what we call friction ridges or dermal ridges that we'll talk about. Take a look at this model though, or look down here. Do you see it? There's vessels that innervate the dermis. There are different types of glands that we'll learn about, different layers of fat, different layers of other tissue. And so we're going to learn how to recognize all of these things by definition and by sight. Thin skin is very similar, except you'll see larger hair follicles as well. So if you can picture that slide you had to learn a couple weeks ago, the hair follicle, you'll see the root of where the hair comes down. You'll see different glands. Your thin skin usually has more sweat glands, apocrine glands, sebaceous glands, oils. And so again, we'll learn how to view these by sight. and by definition. But just as we go through this and as you're thinking about skin, I need you to keep in mind, that we have different layers, different vessels, arteries, veins, nerves that innervate through this and connect to the surface layer. So we'll learn about certain cells that allow us to have touch based on pressure or touch based on an electrical response. And where is that signal going? Well, it has to have in some way connect to that nerve. But it's a lot of these vessels and nerves are very close to the skin, to that outer layer, the epidermis. So here's more of an actual view of someone's skin. Thin skin is going to have, hang on. There we go. Thin skin, hair. Thick skin, no hair. That's not always true, right? There's always somebody that has some wild situation. But on average, thick skin, look at your hands. Look at the bottom of your hands, the palms of your hands. No hair. Look how it's different. Feel that, how it's different. Now look at the back of your hand and your arm. That's thin skin. Think about the difference in feel, the difference in structure with that hair, the difference in the amount of oils and lipids. There's a difference because it plays a different role, different structure. And so again, we'll discuss the different there, but usually if I am asking, a true or false question or a comparison question, hair comes into play as well as depth. So moving into the epidermis, we're going to learn about the cell types first. There are two main cell types, keratinocytes and melanocytes. Remember, O is a linker letter in between two different terms. So keratin is keratin, a protein. Cyte means cell. So keratinocytes probably have something to do with keratin. Sometimes they're called corneocytes after they're fully keratinized. Sometimes when they get filled with this protein, or the protein is around it and makes it this waterproof barrier, they kind of die as our skin moves up and sloughs off. And so there's another word called corneized or keratinized. We tend to stick with keratinocytes in this class because it's more descriptive, but they are also called corneocytes. Melanocytes, what word do you see there? Melanocyte. I'm doing that teacher thing, right? Melanocyte is melanin cell. So these are the cells that are going to produce that thick pigment protein, melanin, that can coat your nuclei and protect from ultraviolet radiation, protect from that damaging ionizing radiation. Give me 1 second. I don't see my cat for 12 hours of a day, but the moment I get on Zoom decides that now's the time to start jumping on my desk or chewing on cords. I don't understand. Anyway, so keratinocytes filled with keratin, melanocytes filled with melanin. Those we could probably pick out. Our definitions or our terms get a little more detailed. So again, maybe you can start writing those flashcards or writing them down, however it is you've been studying. We're also going to come into contact with cells called epidermal dendritic cells. Dendritic means tree-like or branching, and so these are in your epidermis and they have a branching role. Usually when things branch out, they're either needing to be in contact with something else, they're bringing tissue, or I'm sorry, they're bringing oxygen or carbon dioxide to tissue, or they have to be aware. And so in dendritic cells, are cells that have receptors that branch out and play a role in immune response. And so when these cells come into contact with a pathogen, they'll take a part of that protein and they will present it. So in your major histocompatibility complex, your MHC complex, when we learned way back when that you have these special cells that make your body know that you are you, they can present, hey, this piece is not us. What do we do about it? And then, of course, your body can release other cells to destroy those or consume them or take that antigen and make antibodies for it, however our response might be. When you take micro or AMP2, or if you stay with us and you take human physiology, you learn that your immune responses can vary, but these antigen presenting cells present the antigen to say, hey, what do we do with it? So epidermal dendritic cells, usually I always ask about their role in immune response. So if you memorize those two, you'll usually get that question. Now, they used to be called Langerhans cells. In fact, I've stopped calling them Langerhans cells because, again, we're getting away from nominal, I can't, the word escapes me, but the name's based after somebody and more of what they do. So I stick with epidermal dendritic cells. Another cell that we'll talk about is the cells that allow us to have a touch reception. And so we have a tactile response, that means touch response. So we have tactile epithelial cells that'll have little nerve endings due to pressure that allow us light touch. So again, if you have your hands near you, right? And I've seen most of you, so I know most of you do. Look at your hands and then just lightly touch two fingers together and then press two fingers together. Do you feel the difference? Just a small, small difference in light touch, and you can feel that. It's a pressure response when you squish those nerves and squish those tactile epithelial cells. Now, they're called Merkel cells, which is kind of a funny name, Merkel. I don't know, it's like somebody like my sister with her thick accent trying to say miracle. It's a Merkel, but anyway. It's a tactile epithelial cell. Tactile means touch, light touch. And so let's zoom in here. Let's imagine this is a layer of our skin. Here up here, we have our corneocytes or keratinocytes, fully keratinized nuclei. You can't really even tell it's there, but move down, move down, move down, move down, move down. Epidermis. Below the epidermis, we have the dendritic cell branching out, playing a role in immunity. And so this is how the skin plays a big role in immunity. If something gets through there, through a cut or penetration of some sort, and it doesn't belong to us, it's not part of our cells, the dendritic cells are going to try to present that antigen to other immune cells to say, hey, we need to deal with this while we can. Because that's our first line of defense. Our skin is often our first line of defense. Now, if things get lower, that you can have bacterial infections that can overwhelm these, but that's our first line. Move down, look at the tactile epithelial cell. And here you have a nerve ending, a real thin nerve. You have thousands of these right below the tactile epithelial cell that can flood that with a different ion in regards to pressure. And so we can sense that. And over here, we'll have melanocytes producing melanin that can coat our nuclei. And so we learn the cells, good grief, the cells, the layers of the epidermis. We're going to start with the bottom layer of the epidermis and work our way up. Maybe I should eventually reverse this and work from a way top to bottom, but here we are, right? So this is the epidermis. So instead of trying to draw out cells, when I learned this, I would draw them as lines, like we were looking at a geologic time scale. So start at the base of the epidermis. This one is called the stratum. Stratum means that it is stratified. Stratum basale, your base layer. What is the one thing that we're going to remember from our base layer? Here is where the upper layers originate from. Through mitosis, through cell division, the base layer can divide and then provide cells to push the others up. And so the stratum basale is the only layer that undergoes mitosis. The other layers of your epidermis are just those cells pressed upwards. So that is the highlight, circle, star, whatever. The stratum basale is the only cells that undergo mitosis. Above the stratum basale, we have the stratum spinosum, or the spiny layer. These cells look spiky and spiny, so someone called them the spiny layer. It's about 8 to 10 layers filled with desmosomes that pull cells into spiny shapes, so full of these tight weld junctions. These cells are full of RNA, so don't skip ahead. Think. Why or what would it mean that these cells are full of RNA? What would be the purpose? RNA is the instruction for making protein. RNA is read by the ribosomes that make protein. So I bet these cells are doing something that allow the production of a lot of protein. What protein are we dealing with here? Well, we learned that there are keratinocytes. Our skin cells produce keratin, that thick, proteinaceous substance that can fill cells or fill around cells and create this dense, heavy, water-resistant barrier. And so your stratum spinosum is going to be providing the information for that. And as these cells get pushed up, they're going to perform different functions as that hormone or as that protein is produced. So above the stratum spinosum, we have the stratum granulosum, the granular grainy layer, as cells arranged in 2 to 4 layers filled with keratinohyaline granules. Keratohyaline. Well, hyaline is a matrix substance, grainy matrix substance. Keratin is just that protein. And so these are granules filled with this gooey precursor to this keratin protein. And so these layers are going to provide that substance. So they've made in the spinosum, the granulosum, we have a bunch of these granules packed full of this. As we get further up, we have the stratum lucidum. Lucid means clear. So stratum lucidum is the clear layer filled with the keratin precursor ileidin. And so the ileidin is going to become keratin. If we were talking about thin skin, we do not have the stratum lucidum. Stratum lucidum is only in thick skin. The keratohyaline is going to be secreted, and the keratin is going to be secreted to go around these cells. Keratin is just a very thick, hard substance. I'm gonna sneeze, I'm sorry. Moving up a little further, we have the stratum corneum, or the horny layer. I don't know who decides what looks like a spine and what looks like a horn, but somebody did. So they call it the stratum corneum. Corneum is really in reference to the corneocyte. So maybe they should change it to stratum keratin. I don't know, I didn't name them right. Stratum corneum is the most superficial layer. And so at this point, all of your cells, let me go back. As we've worked our way up, so here, epidermis, stratum basale, stratum spinosum, and then we work our way up even further. And so if we're in this layer, no nuclei, that's that flaky layer. Let's go back to the histology slide. That flaky layer, all the nuclei have been pressed out, the skin cells are spread out, they're filled with keratin. Their function is more just protection. And so we say that they're fully keratinized, and that's where they get that name, keratinocyte or corneocyte, depending on where they are. I tend not to confuse you with keratinocyte, corneocyte. In fact, I don't ask you the different name based on where it is, but I need you to understand that as it grows, as it is pushed forward and it fills with that keratin, it is losing nuclei, it's becoming more of a dead cell, just acting as a barrier. This process to go from the basal cell to the corneal cell takes about 35 days. So once every month, your skin cells have rotated. And so you replace those. You know that right now, all the dry skin that we have on our faces and stuff from the winter air, I mean, our skin can flake off and be replaced. This process is controlled by several hormones. I went, and if you have the older PowerPoint from the beginning of the semester, I took a few of these hormones off because it was confusing people. Some IGF and other things that you don't need to know. But we do stick with EGF. Excuse me, I'm so sorry. Clearly today was not, did not want to do this, but we must persevere. Several hormones support this normal growth and repair of epidermis, notably the epidermal growth factor. You can get that right. Which of the following hormones support normal growth of the epidermis? Right? Interleukin-1, insulin, glucagon, epidermal growth factor. I hope that you can pick that one out. So about 10% of all of the cells in the stratum basale are going through mitosis daily. And so they're going through, they are in these big chunks. And so we just divide that into what's called an epidermal proliferating unit, an EPU, and that can be measured. It's usually discussed in reference to burns or wound healing, epidermal proliferating units. And so it's these group of 8 to 10 basal cells that are going through this vertical column of division and then growth as they are pushed up and as they become keratin-filled corneocytes. Below our epidermis is the dermis, but it's not just floating, right? These things have to be connected. And so there's a connection between the two layers. It's a sugar glue, and so it's a polysaccharide gel-like glue that we call the dermo-epidermal junction. Dermis, epidermis junction. So the dermo-epidermal junction is what is connecting, joining those two layers. And let me go back. So down here we have a dermal-epidermal junction. Here we have our base cells, stratum basale, moving up, they can become keratin-filled corneocytes. But go down here where the dermis is. We have our melanocytes and our tactile epithelial cells. Dermal-epidermal junction, we draw as a line here, a glue that connects the two. It's going to act as a seal, a barrier. Yes, some things can diffuse through, but your dermis is fairly deep. Your epidermis is the superficial layer. If you have a wound or a cut or something that has made its way through here, that's very deep. And so that's above our muscle layer. And so we need to protect that. And so we have this dermal-epidermal junction that not only connects these two, but acts as that barrier. Obviously, some things can migrate through, but so the dermis is what we call the true skin. It lies directly below the epidermis and is much thicker. We're not going to learn it in different barriers or different levels. We refer it as, we refer to it as the true skin, and so we don't have to worry about learning different layers of the dermis, but it gives strength to the skin. It serves as a reservoir or storage area for water, water and electrolytes. It doesn't have all of those different layers that the epidermis does. It's much simpler. We have a thin papillary layer and a thick reticular layer. So we don't have to worry about separating it into all those different layers that we did with the dermis. Our thin papillary layer forms what we call dermal papillae. papillae are little tiny bumps, like the papilla on your tongue. It just means a bunch of bumps. And so these dermal papillae actually project into the dermis. And so it's filled with loose fibrous connective tissue and different elastic fibers and touch receptors. And so what they do, if this is your skin barrier, these dermal papillae are kind of branching up in there. So instead of being a flat connection, it allows a joining of the two. And so it just allows for strength between the epidermis and dermis, as well as give more of sensation to touch. And so if we had the tactile cells that do light touch, these larger receptors called tactile corpuscles are going to be dealing with large, thick, intense stimuli. And so that's what's going to allow for more of a stronger pressure response. The thick reticular layer is a dense fibrous network below that. Here's where you're going to find the beginning of hair follicles and these small muscles called erector pillai muscles. Erector pillai muscles pull on the hair follicles and cause piloerection. What does that mean? Think about your hair. You ever had your hair just stand up on end, or you get bumps all over your skin? What do we call that? Well, we call it goosebumps, right? It's a primitive feature. We don't have to use it anymore. So when do we get goosebumps? Well, when we're scared. Yeah, what happens when we're scared? You're seeing an animal, a mammal, when it's scared or nervous, right? Those erector pillai muscles pull, all of its hair sticks up in different areas, makes it look bigger, makes it look angrier, makes it try to protect itself. So it's a very, very primitive function. And then imagine if we were covered in hair. And so think of, other mammals, other primates. When we are cold and we shiver and those erector pillai muscles pull and that hair sticks up, that allows for a layer to collect warm air and that air to sit there and become warmer right next to our skin. So any heat that we have lost gets trapped and warms us up a little bit. Now, we no longer have that amount of hair. We have lost it over the ages. And so evolutionarily speaking, it's not part of our survival mechanism anymore. And so piloerection is kind of just a signal to our past. We still have that response, even if we no longer use that response like it used to be used. And so sometimes we lose features through natural selective processes, and that's just one of them. But piloerection is called goosebumps. This thick reticular layer in animals can be processed into leather. And so you can take this material, you can scrape it, and you can dry the thick reticular layer, and it becomes a very strong, very dense material, waterproof material. And so the erector pillai muscle is this muscle right below on the hair follicle that pulls based on fear, temperature changes or what have you, and causes goosebumps, causes the hair to stand on end. It's what gives you the feeling of, prickling on the back of your neck, right? It's just this response that we still have. It doesn't hurt anything, so it doesn't necessarily give us anything either. So here's our epidermis: stratum basale, spinosum, granulosum, maybe lucidum, it's hard to tell, right? And then of course the corneocytes up here. And so if we were talking about a tactile Merkel cell, that's going to be a real small cell right here connected to a nerve. Look how big this giant thing is, filled with nerve endings. That's a tactile corpuscle. Corpuscle is large. And so this is going to allow for, big pressure. And so when you've been hit or, touched, it's not that light touch response, it's the heavy touch response. And so it's a skin receptor, this large tactile corpuscle in the dermis. Unlike the epidermis, the dermis does not continually shed and regenerate like the I repeated myself, but epidermis goes through that every month, every 30 to 35 days. You kind of, that epidermal proliferating unit has sort of gone through that cycle of mitosis to keratinized. The dermal cells don't do that. During wound healing, the dermis has to form these dense, massive new connective fibers. And so if there is a wound in the dermis, you're going to see that, because it has to like sew itself back together. And so it's not going to do so in that nice smooth area that happened when your dermis formed. And so it's going to scar usually. Usually a superficial wound in the epidermis doesn't always scar, or if it does, it's hard to tell after a while, because those materials are shed, those cells are shed and replenished. The dermis isn't like that. We have what's called cleavage lines or Langer lines. It's patterns, if you were to look at all of our skin, it's patterns formed by the fibers in that particular layer of our dermis as our skin formed. And so all of our skin has been kind of woven in these specific cleavage lines. And so when if you have a good, say we're doing an elective surgery or a surgery and not necessarily, oh no, I have a wound, a good surgeon that is concerned about how this is going to look afterward will try to make the incision or try to close the wound in a way that goes with these cleavage lines. If it doesn't, if it goes against the cleavage lines, you're going to see a massive scar, and it's not going to be held together as easily, because as we move, those collagen move along those cleavage lines. And so this image is trying to show you that if we have a suture along, perpendicular to the cleavage line, that's going to be pulled on, and a scar is probably going to form. It doesn't close as easily. Where if the wound is formed along the cleavage line, you can make a nice clean suture, and when that dermis reweaves itself together and heals through that fibroblast connection, you're not going to see it as intensely. And so for a lot of procedures where they have invasive procedures, they may try to cut along cleavage lines if they can, or if they care, right? And so that's why sometimes things don't leave a mark and sometimes things do, but it's these cleavage lines. Areas of skin connection due to the collagen in the thick reticular layer. So we just discussed the types of cells. Make sure to memorize those. The layers of the epidermis, the layers of the dermis. We're now going to move more into functions of the cell, or functions of the skin, and then talk a bit about appendages of the skin, how those are formed, and then we probably won't have time to get to glands and stuff today, but we'll also talk about glands and disorders and things like that towards the end. We always kind of end with a pathology. So below the dermis, so we have epidermis, dermis. Below the dermis and what is connecting our muscle to our skin is the hypodermis. It's a subcutaneous layer, so subskin layer, called superficial fascia. Fascia is a thin, white connective tissue that's found on top of the muscle. We'll see it when we dissect muscles, but it's located deep to the dermis and forms that connection between the skin and other structures. The hypodermis is not part of the skin. Where do you hear hypoderm? Like hypodermic needle that's thin, tiny, sharp, it can go below the skin. Normally, this is the time of year when I would have everyone look around the room. But think about everybody in the room. Think about your skin color and everybody else. Everybody's skin is a little bit different. Some of us can make freckles, like I can make freckles, right? Little dark patches. Some people have lack melanin. They have very pale, see-through skin. Some people have yellowish skin, olive skin, reddish skin, black skin, brown skin. Why are there so many different types? Well, it comes down to what type of melanin do you produce and in what amounts. There's a genetic component where you're predisposed to the tools that you have available to you, and an environmental component. Our nuclei, remember, every single cell is filled with all 46 chromosomes on average, depending on what you have. And those are ionizing radiation can go in there and break those hydrogen bonds and cause mutations. Things like UV light can cause mutations, and so we have to protect those cells. So what have we developed? We don't have fur. So what have we developed? We developed production of melanin, thick, dark pigment that can coat the nuclei and reflect UV rays. And so it allows protection of our nuclei. We have two different types of melanin called eumelanin and pheomelanin. Eumelanin is true melanin and dark brown. Pheomelanin is reddish and orangish. And everybody produces a different amount of eumelanin and pheomelanin. Melanin is formed from the amino acid tyrosine in melanocytes. So in that base layer epidermis, the melanocytes release melanin packets called melanosomes, and this little packet of melanin moves over to the nuclei. It's ingested by the, it's secreted and then ingested by the keratinocytes, and it forms a cap over it. So melanocytes are not migrating up, but the melanin can be given to the keratinocytes, and it can coat that. Albinism is a mutation where this individual cannot make melanin, and it comes as a range. There's something called leucistic condition or leucism, which is where someone, usually an animal is where we can recognize it, produces very light amounts of melanin, so they look blonde. I always think of squirrels. With the squirrels that we have around here, we can have albino squirrels. They don't produce any melanin. Bright white skin, red eyes. Leucistic organisms are producing some melanin, so they won't have red eyes, but they'll still be light colored. Completely melanized organisms are producing melanin everywhere. And so this is when you see those pictures online of the roosters that are completely black, or the lions that are completely black, or the squirrels that are completely black, because they just produce so much melanin. In humans, albinism comes in ranges, but usually if someone has albinism, their eyes are lighter, they have to wear tons of protective clothing, sunscreen, glasses, even go outside because they don't have that protection and their skin burns easily. Those of us that don't produce a lot of melanin, this time of year we find that our skin is getting very pale if we haven't been outside a lot. Some people can tan, and that just means that they get darker through the increased production of melanin because the sun has triggered that. But you still have this range. Your body can only produce so much based on your genetics. Some people have a ton of melanocytes producing very dark, and it causes that sort of that beautiful purplish, think like Haiti, or what else am I trying to think of? Aboriginal Australian, very dark portion of skin. I'm gonna go down a story. I know I am, and I won't have time for it, so that might have to wait. We'll see how far we get. Let's talk about production first, then we'll talk about the why. We always got to know the why, right? Why do we all produce different types of melanin? And so here is our melanocyte located in our epidermis. And so it's going to produce little packets of melanosomes that are released, right? They're secreted. Look at these little packets. They will be ingested by keratinocytes, brought in through phagocytosis, and then that bursts, and those melanin pigments completely coat the nucleus and stain it. And for that cell's life, for those 30 days, it will be stained with that melanin. Now, tyrosine is the precursor. It is the amino acid that is responsible for it. You don't have to know the entire pathway, but know that tyrosine is responsible for producing melanin, And we each have different types, different amounts of eumelanin versus pheomelanin, reddish versus brownish. Our hair is due to melanin types. And so some of us have darker hair, lighter hair. Some of us, like you've seen my hair, right, have those hair follicles that don't produce any melanin. And so they're just white and clear. But this is just a protective coating of those nucleocytes, of the nuclei. So melanin is processed, the melanin production is regulated by tyrosinase. Tyrosine is the amino acid that allows this. And it's stimulated by the production of UV radiation and certain hormones. We call them melanocortins. These are hormones that control the production of melanin, and UV radiation tends to stimulate the production of melanocortins to say, hey, make more. We're out in the sun. We need to be protected. Sometimes over the years, if you've had a lot of UV rays beating down on your skin and you haven't been wearing sunscreen, it can cause permanent darkened areas that we call age spots. The UV radiation, the reason that we care so much is not only can it cause mutations, it can cause the breakdown of folic acid, and folic acid is extremely important in our health. And so exposure to UV radiation causes melanocortins, as well as the amount of genetics, the genes that determine the amount of melanin that you have. Both of those say, hey, make this dark pigment. And so we can, there's a lot that goes into how that actually occurs, but that pigment can cause darkness. And then skin color is also regulated by the amount of oxygenated blood below the skin and the amount of blood that we can see. And so all that plays a role in the skin color that we end up with. So genes, melanin, blood, essentially. And so we can produce more melanin the more we're exposed to UV radiation. It just, depending on your genetics, depending on your ancestry, you have different types of melanocytes and different amounts available to you. And so if you don't have a lot of eumelanin, you're not going to be able to make very dark melanocytes or very, very melanin-filled keratin cells. However, if you have a ton of that eumelanin, you are going to be able to. And so it's just a protective factor, what has kept us alive. Age spots, we've probably seen someone with age spots before. Maybe some of us have age spots. It's just areas through time where we have darker areas permanently affected by the sunlight. And so that's why if you're every day, you should be using a moisturizer of some sort that has some type of sunscreen in it. I tend to like the ones that have that use metals, little dark metals, and not the chemicals, just because it caused me to break out. But something that somebody taught me last year in A&P and I'd never thought about before is when you know, right before you leave, when you're doing that, putting that on, to take some of it and then rub the back of your hands. Like, why would you do that? Well, yeah, our face is always exposed, no matter the time of year. We also always leave the backs of our hands exposed. And so it's kind of, what's the word I'm thinking of here? It's kind of a vanity thing, right? UV rays, yes, they can cause mutations, but they can also cause wrinkling and age spots, and some things you just can't control, like this lipofuscin production. But it's just one of those things. If you're already doing it right, you know, just a habit. I'm just trying to look out for you, right? That's all. Melanin is not the only pigment. We also have beta carotenes. And what's the word you hear when I say carotene? I think carrot. So carrots are oranges. So we also have some yellow oranges where we convert vitamin A into this orange pigment. And then things called lipofuscins that accumulate, which is what make this age spot. Lipofuscins tend to accumulate in the cell, in cells that have ceased mitosis, and produce that brown yellow age spot because that little packet isn't necessarily going through mitotic proliferation. And so that's why as we age, we tend to get wrinkles, we tend to get age spots, we tend to not heal as well because those cells just aren't going through mitosis as well as they used to. Hemoglobin is the iron-containing protein complex that carries oxygen, and changes in that can also cause changes in skin color. When we have a lot of skin, when we're embarrassed or flushed or hot, when we have it's perfused by blood cells, we see that. Cyanosis is when someone has a bluish color, caused by the darkening of hemoglobin, when it loses oxygen and gains carbon dioxide. So someone's cold, right? Usually their lips or their fingers can become bluish, or if they are not getting enough oxygen. And so that's a sign of choking, it's a sign of poor lung control, or something wrong with perfusion or something wrong with circulation. Cyanotic means this is a sign that, oh no, they're not getting oxygen. And of course, there are other pigments as well from cosmetics, tattoos. Anybody has tattoos, you'll know that, well, how does that stay? How are these tattoos staying in the skin if you're telling me that that cell is going through that constant proliferation? Well, that's because these have been absorbed by those cells that aren't going through that, by those melanocytes, by those other cells that are not going through that reproduction division process. So tattoos and other things that are deep below can cause, if you ever get like a pencil lead stuck underneath your skin, right? That is below that epidermis, or at least that base layer. Where are we at? We're almost done. So cyanosis, cyanotic, always look at someone's fingertips, always look at someone's lips and their nose. If they're cyanotic, whatever, for whatever reason, right? They're not getting enough oxygen. And so that's a big trigger that something needs to be taken care of. Sometimes it happens with age, poor circulation. Other times it's a sign of something wrong with the heart, something wrong with the lungs. Are they choking, When our red blood cells break down, especially under the skin, they release different pigments. And so it can cause bruising when there's damage, bruising when it perfuses an area, and then those begin to break down close to the skin where we can see them. And so as a wound heals, it goes from that dark purplish to yellow as the red blood cells have been consumed and broken down. So let's stop there so that you can focus on all that. Next time we'll go through protection. We'll talk about functions. We'll learn why, the why behind why do some of us have dark skin? Why do some of us have light skin? Why are there different areas of the globe that have people with different colored skin? It doesn't make sense. Well, it does if you can think about what melanin does and how we also have to have exposure to some sunlight. But we'll get into that and then talk about heat loss and then the appendages of the skin. And so we'll cover all that next time. If you have any questions, let me know. Otherwise, I will see you on Monday.